In this video, we are going to be talking about how the non-specific immune response, that's phagocytosis, and the specific immune response, that's T and B cell stimulation, fit together. And we're going to be doing that in the form of doing a giant diagram of the whole process. We're also going to talk about what the cell-mediated response and the humoral response are. And we're also going to be covering what the primary and the secondary response are, as well as going through some exam questions and the type of terminology that is needed for them. This is going to be the best value 20 odd minutes if you're confused about immunity at AS, and especially if you speed me up to 1.5 times speed using the settings wheel below. Be ready to stop and rewind if you need to, as I'm going to hit every specification point and, more importantly, the detail needed in answers pretty darned efficiently. To help even more, I've put up the timestamp so that you can jump between sections. So let's get on with a little bit of context. It's really important that you remember your GCSE immunity. So here we go as a little bit of a reminder. Pathogens are microorganisms that cause disease. And if they infect you, now notice that infection means getting past your natural defences. Your natural defences are your skin, your stomach acid, your tears, your mucus, etc. And infection means that they get past those defences and get into your blood and your tissue fluid. And their actions cause the symptoms of the disease. These symptoms are as a result of one of two things at GCSE, either by damaging your cells, and that is as a result generally of bacterial toxins, or by killing the host cells. And that is often, you were taught at GCSE, as a result of viruses bursting out of cells. Once the pathogen has got into the blood or tissue, i.e. you are infected, then you were taught at GCSE that your white blood cells carry out one of three actions. Number one, you were taught that they engulf and digest the pathogen. Number two, you were taught that a different type of white blood cell produces antibodies. And three, you were taught that antitoxins were also produced. How does this fit into A level? Well, the engulf and digesting of pathogens is phagocytosis. Notice it's a non-specific response. We are going to go through this. The antibodies are produced by B cells, specific response, and the antitoxins are also actually antibodies. The antibodies bind to the toxin and neutralize it, so also B cells. An infection by a pathogen is therefore literally a battle between how fast and how seriously the pathogen can damage or kill host body cells, and how fast the white blood cells can destroy that pathogen or neutralize its toxins. If the white blood cells cannot react fast enough and the pathogen does enough damage to the host body cells, then death is the logical conclusion. If our immune system could not destroy the pathogen for any reason, then we are in trouble. So let's do a little bit of content with some definitions. These will all be clarified again in the diagram that follows. So first of all, phagocytes, T cells and B cells. The T cells and the B cells are a type of white blood cell called lymphocytes, but you can call them T cells and B cells in exam answers. And phagocytes are another type of white blood cell. Antigen, this is a protein or a polysaccharide that generates antibodies. It's why it's called an antigen, because it generates antibodies. It produces an immune response by stimulating the specific T and B cells. Antibody, these are proteins that are specific to a particular antigen. And they are complementary to that antigen. Non-specific immune response. This kind of does what it says on the tin because it is the response that is exactly the same for every pathogen infection. Whereas the specific immune response is a response that is specific to that particular antigen on the pathogen. And it's as a result of stimulation of T and B cells and the secretion of antibodies. And those antibodies are complementary to the antigen. The primary response, this is the multi-step process that occurs from an antigen-presenting cell being detected in the body right the way through to the secretion of antibodies. Again, this will be absolutely clarified in the diagram that follows. And the secondary response is the stimulation of the memory B cells, and they cause the massive secretion of antibodies incredibly quickly after infection, and much more quickly than the primary response. Finally, cell-mediated response. This is as a result of T cells. It's really important that you understand that T cells only bind to antigens on a host cell. That means on an antigen-presenting cell. Once again, I will clarify this in the diagram. And the humoral response is by B cells and antibody secretion. And again, it's important for you to understand that antibodies are soluble proteins. They dissolve in blood and in plasma, and therefore can get to every part of the body because, of course, tissue fluid bathes all the cells of our body, and they cause the destruction of the pathogen. So let's get on with this diagram. Here is my pathogen with a rather oversized antigen on its surface. That pathogen is supposed to be a bacteria, if you hadn't guessed already. And here is my phagocyte with its characteristic multi-lobed nucleus. And I'm now drawing the process that is phagocytosis because the phagocyte has now engulfed and digested the pathogen. But really importantly, it has also presented the antigen from the pathogen on its cell surface membrane. This now makes the phagocyte an antigen-presenting cell. And I think it's very helpful for you to understand that other cells, as well as phagocytes, can be antigen-presenting cells. And that brings about these cells' destruction by the cytotoxic T cells, which we're about to come to. So, antigen-presenting cells, phagocytes, kind of obviously. Infected cells, particularly those by viruses, they will present the viral antigen on their surface. Cancer cells at the early stage of cancer can be destroyed by the immune system, and transplant organ cells, that's why transplant organs are rejected. This whole section here involving the phagocyte is the non-specific response. But importantly, this non-specific response, the phagocyte interacts with the specific response because it interacts with a helper T cell, which I have drawn here. The helper T cell has a specific antigen receptor on its cell surface membrane. Notice that that is going to fit with the triangular antigen. These T helper cells are stimulated by binding to the antigen presenting cell, and that brings about the cloning of this T helper cell and differentiation into a larger population of T helper cells and also the cytotoxic T cells. And these cytotoxic T cells bring about the cell-mediated response in as much that they kill any antigen-presenting cell, those that are deemed foreign to the body. But importantly, the T helper cells also go on to stimulate the B cells. Notice that the B cells don't just have the specific receptor on their cell membrane, they have the specific antibody on their cell membrane. Each of the ends of the Ys of those antibodies have the specific binding site to the particular antigen, the triangular antigen that I'm using. Now, the B cell is stimulated by binding to the T helper cell, but the B cell also has to bind to the antigen as well as the T helper cell. The antigen can either be on the pathogen or it could be free. The antigen is free in the example of a bacterial toxin. The toxin acts as an antigen. And that binding brings about the cloning of the B cell into either memory B cells, as I'm drawing here, with the specific antibody on its cell surface membrane, and importantly, the memory B cells stay in the blood. They're the only thing that stays in the blood, and they bring about immunity. And the B cell, the stimulated B cell, also clones and differentiates into the plasma cell, which secretes the specific antibody. And the antibodies bring about the destruction of the pathogen, or they neutralize the toxin and act as antitoxins. The involvement of T cells and B cells are the specific immune response. But the T cells bring about the cell-mediated response, and the B cells bring about the humoral response because it's involving the production of soluble antibodies. It is the memory B cells and only the memory B cells that bring about immunity. It is only these that stay in the blood. The antibodies do not stay in the blood. They're actually destroyed really quite quickly. Now what I'm doing is highlighting the multi-step process, which is the primary response. And because it's a multi-step process, it takes a long time, relatively. And it involves the stimulation of both the T cells and the B cells to bring about the secretion of antibodies. Whereas the secondary response is a single-step response of the memory B cells being stimulated to turn into plasma cells, and they cause the secretion of antibodies much more quickly than the primary response, and they cause the secretion of many more antibodies. I'm really hoping that that clarifies the whole immunity response. So now let's go on to some exam questions and how to answer them depending on the wording of the question. So first of all, what's an antigen? What's an antibody? So 2-mark questions for each of these. You need to know that an antigen is a protein, but really importantly, a foreign protein or polysaccharide, which stimulates the generation of antibodies. Notice that foreign is the important word there that you might miss. What is an antibody? And notice you need the whole phrase that it is a protein specific to an antigen that is secreted by plasma cells. Secreted does seem to be a favoured term. I would just get used to using it. Second question, when a vaccine is given to a person, it leads to the production of antibodies against a disease-causing organism. Describe how. Notice that the vaccine being given is the start of your answer, and the production of the antibodies is the end of your answer. So you need to talk about the vaccine containing the antigen from the pathogen. Kind of state the obvious. And then you can go on to say that the phagocyte presents the antigen on its surface. Notice that this question is an old spec question. You would not need to use the term macrophage. And the T cell, notice with the complementary receptor binds to the antigen. That stimulates the B cell with a complementary antibody on its surface. And that goes on, the B cell goes on to clone and secrete large amounts of antibody. Notice again that secreting is the favoured term. This next question appears to be very similar, but the start and the end of the question are slightly different. Because the start of the question is again about being vaccinated, but the end of the question is how it leads to protection against the pathogen. And protection means immunity. You need to talk about the memory B cells. So once again in blue, I am going to underline what I think are the minimum simplest answers. So an antigen on the surface of the bacterium binds to a receptor on a B cell. Notice that you would get the mark if you talked about it binding to a T cell. You need to talk about the B cell producing a clone. Don't need to talk about cytokines. That B cell is stimulated by T cells, and that the B cells, and I would personally just jump straight to talk about plasma cells secreting antibodies, but that they also turn into memory cells, and those memory cells cause the secretion of more antibodies. Finally, here's a question that's about how a presentation of a viral antigen leads to the secretion of antibodies. Again, a slightly different question because the presentation of a viral antigen means you're talking about the antigen presenting cell as being the start of the answer. So therefore this answer only involves mark points 3, 4, 6 and 7 from the second question I covered at 17 minutes 10 seconds on the video. But my favoured answers are underlined in blue. Notice that you had to use the term plasma cell. So once again, I would just get used to talking about plasma cells secreting antibodies. And also notice that different wording can be used that means the same thing. I'm Biology Carol, and I hope this video has unknotted the nightmare intellectual mess that is immunity at AS. And it's now a little bit clearer. Don't forget to hit subscribe if you need more untangling, hit like if this has been useful, and leave a comment. I'd love to hear from you.